So good morning, everyone. I'd like to welcome you to our third edition of Proteoform Thursdays. Today, we're happy to have Jana Havlikova with us from University of Birmingham, speaking to us about some of the top-down work she's done on microbes. Let me first give you a brief introduction to the consortium, and then we'll jump in from there. So the consortium is really two things we're trying to do. One is we're trying to build a community to try to help people better interact and build the networks we need to be successful through education, conferences, and things like that. And then we're also trying to develop technology and guidance around what are the best ways to do things, developing methodologies, protocols, through a variety of initiatives, as well as expanding the Proteoform database. We have a variety of resources available as far as top-down standard methods. We have publication database there. We have software tools that are available. These are third-party tools, not things we've developed. We have some guides around nomenclature to help share information, as well as our database that's there as well. So it's free to join the community at this point. You can participate in projects. You can be informed about events and initiatives we're having and just build relationships across the community. So it's free to join, so you can take a moment and please go ahead and do that if you'd like. The next event we're going to have is Charlotte Utrecht to be speaking to us about some native top-down work she's done on viruses and protein complexes. So that'll be fascinating. We'll have more information on that in the next week. And finally, just like to thank our supporters and sponsors, Thermo Fisher, Bruker, and SciEx for being major sponsors of the consortium, as well as Agilent and Pfizer, who are supporting us as well. So before I move to the main part of the program, we just want to have a quick question on what would you folks like to see as far as topics going forward. So if you want to take a moment and just select the topics that you see here that may be of interest to you. And if there's something not of interest on this that you'd like to see us do, just go ahead and put a note into the Q&A, and we'll be happy to consider that as well. So appreciate your input here. We'll let that run for a minute, and then we'll take a look and see, where people's sentiments are these days. Okay, we'll stop that and we'll see. Clearly, data analysis seems to be the top thing. I mean, a lot of people wrestle with that. It's definitely a complex arena. New methods for top-down are important as well. So this is helpful, and this will help us to guide us as we try to look at other topics and other speakers going forward. So thank you for that. And now I'd like to go to the main part of the program and turn it over to Jana, who will tell us about some of her recent work she's done at the University of Birmingham. So go ahead, Jana, I'll let you take over from there.
Thank you. Okay, not the last slide. Is this okay?
That's great, yes.
Okay, thank you very much. So first of all, I would really like to thank for this opportunity to talk here. It's a big honor for me. I'm a very early career researcher. So today I would like to tell you something about my PhD project, which was focused on top-down liquid extracts and surface analysis mass spectrometry of microbes. So first, I will start with a little bit of introduction. So the use of mass spectrometry for analysis of bacteria is currently well established. There are quite a few techniques which are used for bacterial analysis. The most popular tool is matrix-assisted laser desorption ionization time of flight mass spectrometry. This technique is considered as a gold standard, it is used in a clinical practice and FDA approved. However, MALDI-TOF analysis requires sample preparation. The sample is analyzed under vacuum conditions, and a deposition of matrix is also required prior to MS analysis. And these few drawbacks led to development of ambient ionization techniques. So I will show some of them here. This is, for example, paper spray mass spectrometry, desorption electrospray ionization mass spectrometry, or rapid evaporative ionization mass spectrometry. There was also nanodesi or flow probe used for bacterial analysis. These techniques are all great. They have their advantages and disadvantages. But there is one technique in particular that I would like to talk about today, and that is an ambient ionization technique called liquid extraction surface analysis mass spectrometry. Why did I choose this technique was that we wanted to look at bacterial samples without any specific sample preparation, and we wanted to analyze them directly. And we wanted to look at proteins, and we know that LESA is suitable for analysis of proteins. So what is liquid extraction surface analysis mass spectrometry? So it is this robotic platform called Triverse and Nanomane, manufactured by Advion. So LESA is a liquid microjunction surface sampling technique. So what it does is that it has this robotic pipette here, and this robotic pipette picks up a pipette tip from the tip tray. It aspirates few microliters of extraction solvent system, and this is dispensed on the sample surface. In this case, it's a colony, bacterial colony here, in a way that it touches the colony surface. After this step, the extraction solvent system is re-aspirated back into the pipette tip with extracted analytes, and the sample is introduced into the mass spectrometer via chip-based nano electrospray. On the right-hand side, we can see how it looks like. So here is the tip tray, the robotic pipette, the 96 well plate, half of the 96 well plate for solvent storage, and here I have a bacterial colony and mass spectrometry inlet. We've published studies where we use LESA for direct analysis of bacterial colonies from Petri dishes. So in this case, this was a pilot study of investigating E. coli. And then this study, which was published around the time when I started my PhD. And here, 39 proteins were identified by LESA mass spectrometry from multiple species, gram-positive and gram-negative. So first, the main goal of my project was to investigate escape pathogens, identify proteins from escape pathogens by LESA mass spectrometry, and then look into the relationship between the escape pathogens and skin wound infections. So first, I started with top-down LESA mass spectrometry of escape pathogens. So what are escape pathogens? They're a group of microbes which are mostly responsible for hospital-acquired infections. They are clinically important, and their antibiotic resistance is rising. So I've started with analysis of 4 out of 6 escape pathogens. The reason for that was that these 2 species have been investigated in the previous study. So I focus on these 4 species, plus 2 clinically important species or strains, E. faecalis B583 and a clinical isolate Acinetobacter baumannii AC02. So the first thing that needed to be done was an extraction solvent system optimization. So what happened here was that in the previous study, we had 2 different extraction solvent systems for gram-positive bacterial species and gram-negative bacterial species. What was the difference? So here for gram-positive bacterial, we have a thick peptidoglycan layer and a cell membrane here, while for gram-negatives, we have a thinner peptidoglycan layer and 2 membranes. For LESA, it was easier to extract proteins from gram-negative species, and for these, extraction solvent system comprising 40% acetonitrile, 60% water, and 1% formic acid was used. But it wasn't simple to extract intracellular proteins from gram-positive species. So a more harsh solvent was used containing 50% acetonitrile, 45% of water, and 5% of formic acid. I started with analysis of enterococci. These are gram-positive. And I actually, when I've used the 50% acetonitrile solvent, I wasn't able to see any protein peaks in the mass spectra. So I needed to test a range of different extraction solvent systems. So I found out that the 60% acetonitrile, 35% water, 5% formic acid was suitable for extraction of proteins from E. faecalis, and as we can see here, there are protein peaks here in the mass spectrum. I've tested this, of course, for the rest of the investigated escape pathogens, and for E. faecium, we can see that more protein peaks are observed when the 60% acetonitrile extraction solvent system was used. The rest of the investigated escape species was gram-negative, and when I used this optimized solvent system, I could observe proteins in the mass spectra. I manually identified 20 proteins from these 4 species, and then I've also looked at the clinically important strains. So the first one was E. faecalis, and I was able to successfully identify 4 proteins. I've also compared E. faecalis to E. faecium, and I identified 50S ribosomal protein L29 for both of these. The only difference in this protein is this last amino acid residue. So this one is the only difference, and this results in a mass shift. So we were just discussing this, and that this might be potentially used as a biomarker for these strains. So this was interesting. And then the clinical isolate Acinetobacter baumannii ACO2 was quite a difficult one for me because 0 proteins were identified. But the problem was not that I actually haven't seen any proteins. I've seen a lot of them, but probably a new database will need to be constructed for this particular strain. Actually, what was really interesting that when I got these 2 strains, and I was told that they shouldn't be very dissimilar. So AYE is a reference strain, while ACO2 is a clinical isolate. But it's nice to see that with LESA, we can actually see the difference between these 2 bacteria, that when we zoom in, we can actually see that they're not the same. What I've done at this point was that I wanted to see whether my proteins would be correctly assigned if we search against a different database. So I took a tandem mass spectrum, of one of these, I took all of them, but for example, when I take one of the tandem mass spectrum of E. faecium, what I've done is that I searched it against all of the escape protein databases that I've got, and I wanted to see whether it will be misassigned. What happened there is that I took E value as an indicator, the lowest E value as an indicator of a correct assignment. So I've done this for all of these proteins which I've identified, and I found out that 19 out of 24 proteins were correctly assigned, and I calculated the overall success rate, which corresponded to 79%. So just a short summary of this part of my work is that the extraction solvent system was optimized, and this can be used at least for all of the escape species now, regardless of the fact that it's gram-positive or gram-negative. And then I also investigated 2 clinically important strains and compared them to the escape species. And I performed the search against databases from multiple species, and we associated the identification success rate, which was 79%. So as I mentioned before, the first part was focused on identification of proteins from escape pathogens, but then the rest of the focus was also related to skin wounds and infected skin wounds. So I started doing experiments with in vitro 3D skin models. We actually purchased skin models from a company called Innoven. I think they are now LabSkin. And we wounded these skin models with a scalpel blade. And they were all inoculated with bacterial suspension of the desired escape species. These samples were incubated for up to 96 hours, and they were investigated at 4 different time points. So the first one was 24, then 48, 72, 96 hours. And what was really interesting here is that I've tried 2 different extraction solvent systems. So I tried acetonitrile-based and ethanol-based. And what was really interesting that I could extract proteins with the 60-35-5 ethyl water formic acid extraction solvent system. So this was really important for us, because if we want to use this as a kind of like potential diagnostic tool in the future, we want to use a patient-friendly extraction solvent system. So ethanol, I think, is ideal candidate. So this was good. And after 24 hours, I didn't see any bacterial protein peaks in the mass spectra, only skin proteins. So these are actually controls. So this is not wounded, not inoculated sample, and this is wounded and not inoculated sample. So we were able to see skin proteins in the mass spectra. And then after 48 hours, I was able to detect bacterial protein peaks. So as a summary here, so there were 2 proteins identified for each species except for Pseudomonas aeruginosa, but 2 additional proteins were identified after 96 hours of incubation. So what was really interesting as well here is that so after 48 hours, colonies were formed inside the wound. So here is a colony, and this is a wounded sample, and this is sample which is basically a 3rd type of control. It's a sample which is not wounded but inoculated with bacteria. And what happens here, so especially for Pseudomonas aeruginosa, that was really specific, was that here there was a significant damage of the skin model. So basically, the skin model was non-existent at the end of the experiment. And then what is even more interesting is that there was no damage observed in the control sample. So we basically had the inoculated sample, but not wounded, but they just behave as non-inoculated samples. So there was no damage. But this kind of this makes sense, because you can have pathogenic bacteria on your skin, but unless you have a skin wound or some scratch, they should be fine. So this was really interesting. So a summary of this experiment is that we used a patient-friendly or ethanol-based extraction solvent system, and we're capable to extract proteins from human skin and also bacterial proteins from our samples. And we'll also we're able to see bacterial proteins after 48 hours of after inoculation. But the very important thing is also that we were able to see both bacterial and human skin proteins in the same mass spectra. So for the future, this is important, because we might be able to look at possibly at human or host pathogen interaction or the response of the host. So these experiments led to this time's analysis of ex vivo human skin. So one of my supervisors, Robin, he was involved in project which allowed me to collect skin grafts. And he's also suggested to look at this study, so maybe we can do something like this. And what happened here is that the skin grafts were sutured onto a cell strainer. And what was important here is that they were cultured in a medium, but then also they were maintained at an air-liquid interface. So this was very important for skin because normally you don't want your skin to be wet. And the second thing is that there was this suturing part which kept the skin tension. So it was much better when these grafts were sutured onto skins onto the cell strainer. And I developed my own workflow, which is shown here. So I took a little piece of a skin graft, I used a surgical needle, I sutured it so it looked something like this, and then they were cultured in a typical DMEM cell culturing medium, and again analyzed in 4 time points after 24, 48, 96 hours of incubation. So it looks something like this. But what the result was, that's shown here. So I was able to obtain a mass spectrum which contained proteins. And then I compared this mass spectrum to the lab skin mass spectrum, and I could actually see that there were the same skin proteins observed. So this one was a ubiquitin, and this one was an unknown skin protein. So I did fragment it, but I couldn't really identify it. So I couldn't get the ID. And I also had another problem with human skin model, with the human skin grafts. And that was that because they were cultured in that DMEM medium, they were full of salt. So it was not very good for mass spec. And I tried the several washing protocols, but that didn't really help. So this was something that I could see. And I was still able to fragment these 2 protein peaks. So one was identified as ubiquitin, and the second one was identified as S100A7 or psoriasin. So both of these were observed in the lab skin model mass spectra and identified. So at this point, I had to stop. Well, the work was disrupted because of COVID, and then after the first wave, when we're allowed back into the labs, I didn't really. It wasn't really a good idea to work with the human skin samples, or nobody was really thinking about it. So this work was disrupted. But what I'm actually able to say is that the skin grafts were successfully cultured for at least 96 hours. And the mass spectra that I obtained were similar to the lab skin mass spectra. And I was also able to identify 2 human skin proteins. And now I would like to talk about the most recent work, which was done on the thermal Orbitrap Eclipse. And this was really exciting when we got the eclipse. So the first sample I wanted to analyze was the E. coli K12. So it's the proteome of E. coli is very well annotated on the UniProt, and it's a great model organism. So first I've started with a comparison of the agar background and E. coli K12 colony. So as we can see here, so here in the upper mass range, there is quite a difference, but here I just want to look a bit closer at this area. And what we can see that here is a typical agar background signal, so no proteins, no peaks corresponding to proteins are present here, but in the bottom mass spectrum, which is the actual E. coli mass spectrum, we can see many peaks corresponding to proteins here. So it's very busy, and that was really exciting. Then I decided to compare the mass spectrum that I obtained on the Orbitrap Eclipse to the mass spectrum from the Orbitrap Elite, and there is a huge difference here in the lower mass range. So normally the Orbitrap Elite, we don't really see many proteins in this lower mass range, but here we could see many of them, and they are actually peaks corresponding to proteins. So I started with manual protein identification, but after a very short time, I found it very overwhelming and time-consuming because there were too many proteins, which was really good. But for example, these 8 proteins, I identified them in a very short time. So it was very good, but I wanted to identify. So the goal was to identify as many proteins as I can in the shortest time possible. And we at this point I would like to start talking about FAIMS. So FAIMS came in at this point because we thought that FAIMS can improve. Not we thought, we have actually seen that FAIMS can improve the number of identified proteins. So for those who don't know what FAIMS is, it's a high field asymmetric waveform ion mobility spectrometry. So you got the new FAIMS Pro device, which is shown here. So the FAIMS Pro device is attached to the Orbitrap Eclipse. And this is the FAIMS device, and it is located between the LESA platform and then the mass spectrometry inlet. So what how it works. So I wouldn't extract the proteins from bacterial colonies as I normally would, and then introduce them into the mass spectrometer by nanoelectrospray, the chip-based nanoelectrospray. So now we have ions in the gas phase, and here we have the FAIMS device. So the ions will enter the FAIMS device. And what we know is that the mobility of ions is different in high and low electric fields. So basically what happens here is that an asymmetric waveform is applied, and the mobility of these ions is different in high and low electric fields. So what happens is that they would when the waveform is applied, the ions would deviate from their original trajectory, and they would eventually collide with one of these electrodes. But what we can do is that we can correct for this deviation by applying compensation voltages, and by applying different compensation voltages, we can selectively transmit ions through the device. So when we apply one compensation voltage, we would transmit the red ions, with a different one, the green ions, and so on. I want to point out here that, so we know that FAIMS actually does help with or increase the number of identified proteins, and we've published a study on this before, but it is important to say that in this study we've used chip-based FAIMS device, not this cylindrical FAIMS Pro device. So at this point, I would like to call this high throughput protein identification, because we were actually, this was the goal, that we wanted to identify as many proteins as we can in the shortest time possible. So I've got two different data sets. So I've always analyzed my sample without FAIMS device attached, and then with the FAIMS device attached. So I investigated 6 different CVs. So I started with minus 10 volts, and I ended at minus 60 volts in 10 volt steps. So I got 6 data files for FAIMS data and 1 file for no FAIMS data. So this instrument method was developed. So I developed, it consisted of 2 steps. So the first step acquired data for half a minute, so that was a control, and the second step used data-dependent CID MS, and I used top 10 method. The whole method was acquired for 10 minutes per CV, or 10 minutes when no FAIMS was attached. The step 3 included database search in ProSite PD 2.4, and all files were searched at once with 1% false discovery rate for FAIMS data, and the no FAIMS data were searched separately. So this is table, this graph, sorry, is a kind of summary of the number of proteins identified for these 5 investigated bacterial species. So the blue bars show the manual identification, the orange bars represent high throughput no FAIMS identification, and then green bars high throughput FAIMS. As we can see, it's a significant improvement in the number of identified proteins with the Orbitrap Eclipse in general, but then with FAIMS, it is even better. And with the manual identification, that would take days to identify this amount of proteins, and now with FAIMS we can do it in 1 day. So that's a major improvement. I've also looked at the number of proteoforms identified with and without FAIMS. So for all of the bacterial species investigated, I could actually see a higher number of proteoforms when the FAIMS was attached on the Eclipse than when it wasn't. I just want to say that I miss error bars here. It is because because of the COVID disruption, I wasn't really able to do a number of replicates. So I know that the error bars are missing here. So just to let you know. So we have some interesting observation here. So while I was looking at these 5 bacterial species, what I've seen was that when I applied less negative CVs, minus 10 volts and minus 20 volts, there were some kind of high molecular weight proteins observed in the mass spectra. And this was very unusual because I was using LESA and denaturing extraction solvent system. So what was usually observed with LESA and this denaturing LESA is that the kind of like we were usually seeing around 10 kilodaltons up to 15 kilodaltons of molecular weight of proteins of this molecular weight. And suddenly I've seen like something really big there. So that was really surprising. And for example, for E. coli, when I applied a compensation voltage minus 10 volts, I was able to see this 27 plus protein peak here. And so what I've done is that I use HCD fragmentation in this case, and then I manually collected the data and identify this protein as an outer membrane protein C. It was almost 39 kilodaltons. Then for Klebsiella, I've seen this protein, and it was identified as an outer membrane protein A, almost 36 kilodaltons molecular weight. And then the last one, which was above 30 kilodaltons, was this protein detected at compensation voltage minus 20 volts, and it's a probable binding protein component of ABC transporter, and it is a D-amino acid transporter, and it was just about 30 kilodaltons. And I was very surprised. I mean, the sequence, there were actually many fragments there observed. So just a quick summary of the last work, which was done on the Orbitrap Eclipse. So I was capable to identify many proteins from the mass spectra acquired on the Orbitrap Eclipse, which was kind of expected, but it was a good thing to see. And then we've also used cylindrical FAIMS Pro device, which helped us to significantly improve the number of identified proteins in a short time, which was in this case 1 day. And then we were also able to see high molecular weight proteins about 30 kilodaltons for 3 bacterial species with the use of FAIMS device. So I would really like to thank my supervisors, Helen, Ian, and Robin, and whole Cooper group, and also Lini group and past members of the Cooper group, Claudia, Eva, Rian, Happy Lab, in particular, Emma and Farhana. And I'd really like to thank EPSRC for funding and Sci-Fi Center for Doctoral Training for letting me to do a PhD in the UK. And of course, thank you for listening.
Hey, thank you, Jana. It was very interesting, that tour of how you evolved the methods from start to finally having a high throughput approach. So if folks have questions, you can type those in the Q&A, or if you'd like to ask your question live, just go ahead and raise your hand and we can unmute you. I just had a quick question. When you mentioned a number of proteins you couldn't identify, do you know what was leading them to be unidentified? Is it they're just not in the database, or the data was poor, or noise, or what kind of factors caused it to not be identified?
Sometimes it's probably the database. Sometimes it's probably the data as well. I would say that mostly it was, I think, a database problem. But yeah, and most of the proteins that I've actually we're picking up with LESA are uncharacterized proteins. So probably if there are like some proteins which haven't been inferred from homology, or like how to put this? Yeah, so they are uncharacterized, so we don't know what they are. They wouldn't be present in the database because, like, for example, E. coli is very well annotated, but the rest of the species, it's not that great. So yeah, the database would be probably the problem.
Okay, there is a question that came up on the chat first. It's from Zhao Dong. What are the major differences between wounded and healthy human skin on mass spectra?
There was a difference in one particular protein, and that was that we've observed, and that was beta defensin. I think it was like, well, wounded and healthy. Do you mean like wounded, just like wounded, or wounded and infected? Because this actually makes a huge difference.
I'm not sure. I was just reading the.
Yeah. I
there was
see.
additional comment there.
Maybe if the person who's asking could specify, because if it's only wounded and healthy, like a control, there might be some difference. But because I'm not doing quantitative, I can't really say. But when it comes to wounded and infected and then healthy, then yes, there was a difference, especially for one protein called beta defensin, and it's an antimicrobial protein, and we saw definitely like less abundant. This was less abundant in the infected samples than in the healthy samples. But again, because I'm not really doing quantitative, I can't really say what was the difference.
Okay, well, thank you. There's a question from Moe. It says, great work. Similar question about the unidentified proteins. have you tried to de novo sequence or use different search algorithms to get a better idea of what they are?
Yeah, de novo sequencing is actually really difficult because it's, well, I've tried a little bit, but the problem is that this sample is analyzed directly from the colony, so it's very complex. So actually, I think that Dina was, I mean, the previous colleague who published the previous study, she's done that, and she successfully identified one protein. But I think like it would take so much time to do this that we would rather just have less proteins identified, and we would rather move to right now to a high throughput approach, so we can, because what we want to do is, our aim is not to identify new proteins, but to identify as many as we can in the shortest time possible.
Thank you. Question from, excuse me if I missed Manasi. Thanks for the interesting talk. How was the experience of using ProCyte? Any suggestions for other open source algorithms?
I haven't really used any open source. I think there is MASH. I haven't really used anything else than ProCyte BC and ProCyte BD. Oh yeah, yeah, there was another comment that popped out. It was topic. Yeah, I heard about this, but I haven't actually tried. I've used ProCyte BC most of the time, and I think it works. I think it works well for what we do. I think it works really well.
Okay. Thanks. So Joe Lu has a question. Does the use of denaturing extraction solvents affect time course studies? Wouldn't acetonitrile and formic acid negativity this work and require a new sample to be measured?
Okay, time course. So yeah, so I use this extraction solvent system, and I don't think there is like, so I always, when I do like the new extraction, like for each sieve, I don't know exactly is this related to, it's probably like, oh, you mean like with the skin, or? So I can actually, like, if this is related to the skin work, like the lab skin work, I could actually see where the skin was sampled. So I usually try to choose a new location, and this is for all of the samples. So it's actually visible on the skin where you have sampled before. So I was always trying to move to a new location, so it doesn't really affect the number of extracted proteins.
A question from James. I don't know how much time the proteins spend in the extraction solvents, but have you observed any side reactions from Fischer esterification with the ethanol formic acid? Also, what is the rationale for searching only minus 60 CV to minus 10 CV with FAIMS?
Okay. So the answer to this question is not a lot of time. So it's probably a matter of minutes, like here. It's like, so for example, for the Eclipse work, that was 10 minutes. And also, so the thing is that I kind of can't really spray it for more than 10 minutes because, I mean, there were cases when you could have a very long spray going on, but usually with bacteria, it can really clog the small nozzle on the chip, and it's really tricky sometimes, but you can't really spray it for a very long time. So I don't think it's really affected from, like, it doesn't really, yeah, I don't think like there were any side reactions observed. At least I haven't. So the rationale for searching only minus 60 CV to minus 10 CV. So what you can do before your experiment is a CV, it's called a CV sweep. So you basically select, I don't know, you start at minus, I think it was minus 100 volts, or definitely minus 80. And then you go also towards positive voltages. And you can actually see at which point you see the most of your signal, or if that makes sense. And then that was why I chose this. And I also couldn't really see any proteins or identify proteins beyond -60 or at positive voltages.
So we have another question in the chat from Xiaodong. Without much cleanup of the samples, what is the dynamic range of this liquid extraction surface technique?
What do you mean with the dynamic range?
I don't know, perhaps high and low levels of proteins that you're seeing. What's the differences in amplitudes of signals?
Can you please specify? It's just the person who's asking. If you just specify, I'll answer that.
Okay, so would that maybe have some more comment on that, or raise your hand if you'd like to ask live.
Yes, please.
In the meantime, Jolu has a question. Would the LESA method work if you wanted only to identify surface bacterial proteins as well as secreted proteins?
Yes. So you can definitely look at only well, you can look at like secreted proteins, because what I normally do is that I touch the colony surface with the pipette tip. And for example, you don't really have to do this. You can only kind of dispense the solvent on the top of the colony and we aspirate it back. And this actually works for Staphylococcus aureus, which has this delta hemolysin as a secreted protein. So you would be able to see only that protein. But when you actually touch the colony surface, you are able to see intracellular proteins.
Great, thank you. I just had a question. I mean, obviously this is aimed at doing something clinically important someday. So what do you think are the next steps to move it more towards the direction of being enabled in a clinic? Is it just identifying specific markers or refining this method to be more robust? I'm just curious, kind of what do you see going forward?
So I think like the high throughput identification was a very good step. And then also like probably improvement database searches, and also like I think like people were voting at the beginning of this seminar for like more the computational analysis for top down. I think this would be the way to go in order to, I think, make it more robust, even more robust.
Mm-hmm. Great, thank you. Any final questions or comments?
So with the dynamic range, I mean, I don't. Yeah, I don't know. If you can specify, please, that would be great. Or just contact me. That's fine. I can talk about this.
Okay, great. So I think we'll close there. Thank you, Jana, for interesting presentation on the applications of top-down. It's different from a lot of the basic biology we've looked at, and to see how real-world applications this could be really important. So thank you for this. We really appreciate your time and your effort, and thank you everyone for joining us. As I mentioned, July 15th will be our next seminar. We'll have more information going out next week on that. So please keep an eye out for that. Thank you all for attending, and have a great day. Thank you. Bye-bye.
Thank you.